Hello, everyone, and welcome to our webinar on mastering DNA polymerase selection, tailoring your enzyme for PCR success. My name is Gintaira Garoitita, and I am a product manager at Thermo Fisher Scientific. Today, we will explore the essential aspects of selecting the right DNA polymerase for your PCR applications. Now let's take a look at the agenda. We will start with an introduction to PCR, covering its basic principles and components. Then we will dive into the important features of DNA polymerases and the products used for PCR. Finally, we will discuss various PCR applications and considerations when choosing the enzymes. Let's look at the components of PCR and how it actually works. The standard PCR reaction includes DNA template, which is a sample DNA that contains the target sequence. DNA polymerase, this is an enzyme that is a key to PCR. It uses single-stranded DNA as a template to synthesize the new complementary DNA strand. Also, primers, it is a short from 18 to 30 nucleotides long synthetic single-stranded DNA sequences that are complementary to a DNA sequence of interest. Each primer has its own specific annealing temperature, which is crucial for the specificity and efficiency of the amplification. And deoxyribonucleotides, or DNTPs, are used by DNA polymerase to build the new DNA strand. The reaction also includes a reaction buffer containing salts and buffering system. Magnesium ions are always present in the buffer as they are essential cofactors for DNA amplification. All these components are required to perform PCR in a thermal cycler, which principle you can see on the right side. The process begins with DNA denaturation at 95 degrees Celsius, followed by primer annealing, and finally DNA polymerase extends the second strand. This cycle results in exponential amplification of the target DNA to generate specific PCR product. PCR can be classified into different types based on the analysis of the resulting product. In the endpoint PCR, the amplicon is analyzed at the end of PCR by running agarose gel electrophoresis. This is a good approach if the presence and size of the PCR product are of interest or if there is a need to purify the PCR product for any further use. If there is a need to quantify the amount of initial DNA, quantitative PCR can be an option. In qPCR, the product is quantified at every cycle. In digital PCR, the sample is separated into many individual partitions and target DNA molecules are directly quantified. This method ensures excellent sensitivity and accuracy. Now let's briefly talk about the essential PCR components, DNA polymerases. They possess several critical activities that ensure accurate and efficient DNA amplification. First one is polymerase activity, which allows the DNA polymerase to add nucleotides to the growing DNA strand in the 5' to 3' direction. The second one is 3' to 5' exonuclease activity or proofreading activity that allows the DNA polymerase to remove incorrectly incorporated nucleotides by excising them in the 3' to 5' direction. And the third one, 5' to 3' exonuclease activity. The activity allows the DNA polymerase to degrade nucleotides in the 5' to 3' direction, which can be used to degrade probes like TACMan during the PCR process. It is important to note that not all polymerases possesses the same activities. For example, Taq polymerase has only the 5' to 3' exonuclease activity, while proofreading polymerases have the 3' to 5' exonuclease activity. Successful DNA amplification depends not only on the intrinsic activities of DNA polymerase, but also on various other critical characteristics of the enzyme. When selecting a DNA polymerase, the following characteristics should be considered. First one is specificity. It's an enzyme ability to amplify only the intended target without off-target amplification. Another one is hot start capability. The enzyme is inactive until heat activation. Also, fidelity, the accuracy of the polymerase incorporating correct DNTPs. Processivity, the ability to synthesize long stretches without dissociating from DNA, which affects reaction speed, inhibitor tolerance, and amplicon length. Other important factors include DUTP tolerance or the ability to incorporate DUTP, GC-rich sequences, amplification, and buffer compatibility. Each of these characteristics can significantly impact the performance of your PCR, so it's crucial to choose the right polymerase for your specific application. Some of these characteristics will be discussed in the examples in the following slides. Let's delve deeper into the concept of enzyme control through hot start mechanisms. Standard or non-hot start enzymes are active at room temperature during reaction setup. This can lead to amplification of nonspecific targets from misprimed oligos. Consequently, this can result in decreased yields of the intended targets and lead to reduced sensitivity that you can see in the figure in the bottom right, lane 2 and 3. Of course, you can set up the reaction on ice, which will chill the polymerase and lower its activity, but can deactivate it fully. A better approach is using a hot start enzyme, which is modified to be inactive at room temperature, typically by antibody, chemical modification, antibody molecule, or aptamer. After heat activation during the initial heating step, the enzyme becomes active just in time when it needs to synthesize new DNA strands. Hot start enzyme helps provide better specificity and yield. Now let's move on to discussing specific examples of polymerase selection for various applications. Selecting the right DNA polymerase is crucial for the success of your PCR experiments, and different applications may require different enzyme characteristics. If you just need to detect target sequences by their presence or absence, a tag-based enzyme like platinum tag hot start DNA polymerase is ideal. Typical applications of a standard PCR include high throughput PCR screening, genotyping, sequence detection, and colony screening. Here we will give an example of genotyping experiment. Genotyping is used to detect gene variations in alleles in specific cells or organisms. Tag-based polymerases like platinum 2 tag offer specific and sensitive separation of alleles based on the length of amplified products that are crucial for identifying genetic variations. If you are looking for accurate DNA amplification, high-fidelity polymerases are crucial. These polymerases are typically recommended for applications like cloning and subcloning, template generation for sequencing, mutagenesis experiments, and genetic variability studies. Here we present a cloning application, which is an amplification of the desired DNA sequence and its insertion into specifically designed compatible vectors. High-fidelity polymerase, like in detergent platinum superphite 2, is recommended for these applications because it provides high accuracy and yield essential for proper clone insertion and expression. As mentioned previously, applications such as cloning require proofreading polymerases that ensure DNA amplification with high accuracy, minimizing errors in the amplified fragments. The terms accuracy and fidelity refer to the number of incorrect nucleotides that a polymerase incorporates into the newly synthesized strand, and these errors are subsequently propagated in all subsequent copies. While all proofreaders show a higher fidelity than standard tag, the difference between them lies in how much more accurate they are. The fidelity of the enzyme is typically measured by next-generation sequencing and normalized to tag DNA polymerase, as illustrated in the graph below. While some proofreading polymerases have just an 8 times higher fidelity, newer proofreaders like Superfy are over 300 times more accurate, resulting in fewer than 1 in 10 clones containing errors in cloning applications. The direct PCR approach allows DNA amplification without prior DNA purification, significantly reducing workflow time. It is especially useful for small samples as it requires minimal handling and reduces the risk of sample loss. This makes direct PCR perfect for applications such as genotyping, GMO detection, or food and water testing. In this example, we demonstrate bacterial contamination testing by amplifying bacterial DNA directly from lake water samples. Direct PCR from crude samples requires inhibitor-tolerant reagents such as Platinum Direct PCR Universal Master Mix. It ensures high specificity, yield, and persistivity for your PCR experiments. In the previous slides, we talked a lot about polymerase characteristics, and now I want to show you some additional PCR characteristics that should be considered if you want to get the best PCR results. The first thing to be aware of is buffers. They ensure optimal conditions for DNA polymerase activity and even improve PCR. Improved PCR can be achieved by changing salt composition to enhance primer binding specificity, adding chemical additives to improve amplification specificity and efficiency, also adding PCR reaction enhancers for difficult templates, for example GC-rich templates, or simplifying detection step by adding dyes for direct sample loading. One unique advancement Thermo Fisher Scientific offers is the universal annealing feature. The recommended melting temperature of PCR primers is usually in the range of 55 degrees Celsius to 70 degrees Celsius, which can vary due to differences in primer sequence, length, and composition. This variability necessitates time-consuming optimization of annealing temperatures. To address this, we have developed novel polymerases with reaction buffers containing an isostabilizing component, allowing for a universal annealing temperature of 60 degrees Celsius. This innovation also enables the simultaneous cycling of shorter and longer amplicons using the same extension time and allowing different PCR assays to be run together using a single protocol. Another important thing to consider is uracil-tolerant polymerases. Uracil-tolerant polymerases are specialized enzymes designed to amplify DNA templates containing uracil, which can be challenging for standard polymerases. These polymerases are particularly useful in several specific applications like amplification of bisulfite converted DNA, amplification of damaged or ancient DNA, where cytosine residues may deaminate to uracil over time and carry over contamination control when using the UTP containing mixes. Typically, in PCR setups, incorporating the UTP instead of the TTP in the reaction mix and using uracil DNA glycosylase can help prevent contamination from previous PCR products. To further assist you, we have several resources available. The Molecular Biology Education Library, which provides information on various molecular biology techniques and applications. The PCR enzyme selection tool, which can help you choose the right DNA polymerase for your specific needs. Information on the Platinum Superfy 2 DNA polymerase, including detailed product specifications and application notes. And additional PCR Basics webinar, which cover various aspects of PCR and provide practical tips and insights. Thank you for attending our webinar. We hope you found it informative and helpful. If you have any further questions or need additional support, please feel free to contact us.